The Institute for the Study of Pig States presents the 5th International Psycho-Immunology and Psychobiology Research Symposium. In this presentation, Phase 2 clinical trials using psycho-immunology by Dr. Kirsten Lykkegaard.
So our next speaker is Dr. Kirsten Lykkegaard. She's our Director of Research at the Institute for the Study of Pig States. She's trained as a vet and has got a significant experience in pharmaceutical research. So hand it over to you, Kirsten, to talk about some of the phase 2 clinical trials you've been doing recently.
Okay, thank you. Yes, so I'm very excited today to actually share with you what we have been doing for the last about 7 months now in our phase 2 clinical trials, where we have tested the new psycho-immunology treatments that we have. So let me start with, if we just knew everything, then everything would be good. But the problem is that we don't. So you don't know what you don't know. And that's what Mark Twain says. It ain't what you don't know that gets you into trouble. It's what you know for sure that just ain't so. So with that, I'm going to lead into that. When you think of drug development, and you can wonder, why do we even need clinical trials? Why do they exist? So most people probably don't know what happens behind the scene in the drug development. And when you kind of touch at it, you can just definitely see that clinical trials are necessary. Because again, you don't know what you don't know. And this is a pioneering area, which means you're going into uncharted territories. And I have an example from my own life, because I was at a scientific meeting. And I just vividly remember we were in a meeting and we came out in the hall, and then all this buzzing. The news had just broken that a phase clinical trial in London had turned into a nightmare. This was in 2006. And this was a drug that was, it was first human dose. So 6 healthy men were injected with 2 minutes in between with this immune modulating drugs. And the company had really wanted it out there because it was going to be good for leukemia and rheumatoid arthritis. So really needed. But these young men, they all collapsed. They had cytokine storm. Their organs were failing. They went into emergency room within the first 16 hours of this after injection. They all survived, but it was just really an eye-opener that you just don't know what you don't know. And so we all have assumptions, but we really have to check in real life to see if they're valid. And in this case, with this study, they had done a pretty good job. You can always criticize with looking in the rear mirror and what you have been doing, but they did preclinical studies in monkeys, so in primates, the closest you can get to human beings, and they had seen nothing. They were at a dose that was 500 times higher than these 6 volunteers were injected with. So just respect to this feeling of you think you know, but you really don't. And here's the assumption again, because now we walk into what we're doing, psychotherapy and trauma healing. So nothing can go wrong, right? Because we have these assumptions that there are no drugs or no surgeries. So it'll be all right. And all things we do, psychotherapy, trauma healing, that's intrinsically safe. And personal growth with psychological healing, it's always a good thing, right? Seems reasonable to have these. But in reality, because we, with our stuff, psychoimmunology and psychobiology, we bridge the psychology-biology divide that they're separate. In reality, psychobiology and psychoimmunology techniques, they physically modify the interior of the cell. And in some cases, this might trigger if you're not aware what you're doing, unexpected physical, biological, and even subcellular symptoms. They can also expose emotional issues, and in rare cases, a person can go into spiritual emergencies. And with our work, we just don't want that to happen with our treatments. So that's why we do what we do. So ISPS safety testing, we have this, we just want our treatments to be safe and efficacious. So we safety test, and we safety test all new psychotherapy techniques, simple or not simple. So psychotherapy techniques, psychology techniques, and the newer psycho-immunology disease treatments. And like Shane said, we're not regulated by any medical authority, but we just feel that this is the right thing to do. We want to be providing safe and efficacious treatments. We also want to know in here, are there any specific problems so that we can, if we cannot avoid them, if they're intrinsic to the treatment, for example, we know how to prevent them or counteract them as part of the treatment. So again, we just want to know what we might not know to begin with. So sort of figure it all out so we have a good map of what we're doing. And we want to provide easy to do treatments so that most people will be able to perform them. So that's all part of what we do when we safety test. So let's just take a look at what kind of levels of safety testings, because there are, just like in drug research, we start with what in drug research is called preclinical testing or non-clinical. And that's basically where you have an idea, we want to do this, and then we, in the basic research staff, 2 to 3 people do this work on themselves. And if we don't have what is needed, say it's a disease, then we work with someone volunteering to do so at this stage. So that's the first part. Once we have what we call proof of principle, that means we know that when we do this, we are aiming at the right thing and we have it's working well, then we go into phase 1 clinical trial. And this is where we have a group of volunteers, 6, that are a group that we work with on a constant basis here in the Institute. They are certified therapists and they're very dedicated to move these treatments forward. So I'll come more. They'll actually have they'll have a talk tomorrow and share with you how that work is going and what it's like to be in that test group. So we have phase 2 clinical trials. That is trained certified therapists that are volunteering their afternoon, evening, or whatever to do these tests with us. And we aim for having somewhere between 12 and 20 in each of our test rounds on each process or each treatment. After that, if we should just continue this comparison to medical drug development, phase 3 clinical trial would be the equivalent of what we do when we release cautiously to our clinic staff to do these treatments with real clients or that has the underlying disease or condition. And it's in a controlled and closely monitored setting. So this is why we don't just release these techniques directly to the therapists. What I'm going to talk more about is this phase 2 clinical trial in this talk. So as Shane said, I have another slide before that. By phase 2, we actually believe that there are no safety problems. We have pulled all these out before we go into phase 2. But like I've said earlier, we just have to test to be sure because you just don't know what you don't know. So the problems that occur with trauma therapy also occurs with psycho-immunology, and that's because we use trauma therapy. And that means if a client doesn't finish a process that you have started, what happens? Well, they have now triggered some emotions that they haven't finished healing, and those emotions will just fade slowly over time. And this is normal. That's the normal risk that you can inform your clients when you do trauma therapy. We all have that as as an outcome of doing that or as a risk factor. Just a caution. The other good news, because I think all this is good news, that we're in this category of what we deal with. The other good news is that we actually, with these new processes of psycho-immunology, we don't think that there are any adjustment issues. If you look at the old silent mind technique, the one that removes the voices, after doing that, some people would have loneliness. So again, this is an adjustment issue. All of a sudden, their voices are gone. With this new way of doing psycho-immunology, these processes are designed so that this will not happen. And so far in our testing, this has been true. So that's a good thing. So let's look at the current phase 2 clinical trials. When we started this, we actually chose the 6 most common pathogen-caused psychological issues that therapists encounter. And it's also the ones that cause the most havoc in clients' lives. So I think Shane might have mentioned a few of them. So we have Borg fungus. I'll have details on these in the next few slides here. So the Borg fungus, manipulation fungus, we have something called S-holes or sucking holes. That is a pathogen, and we have a repair also included in that process. We have a process for copy bacteria, fine cone fungus, and soul loss. So those 6 most common pathogen-caused psychological issues, we train our therapists in doing this, and we train them in the psychobiology version of how to do with it, how to deal with this on a 1-by-1 fix. And we will continue doing that in our training because this is really crucial that therapists know how to do this on a 1-by-1. It's not just removing them all, because sometimes you just want to go fast on one symptom in a client, and then the 1-by-1 can be ideal. So you want to know the basics of what everything, the biology and what to do in each situation. So, but the PI is a global fix, psycho-immunology, and it eliminates the pathogen completely out of our cell. And with that, all symptoms caused by this pathogen are immediately eliminated. It's awesome to see. Just like Shane said, you can have the symptoms, and then when the healing is complete, the symptoms just vanish in front of your eyes. And this fix is permanent if the process is completed. So let's take a look at silent mind caused by the Borg fungus. This gives a cluster of symptoms. And if you eliminate the Borg fungus, the following symptoms are eliminated. Voices, also called involuntary or obsessive thoughts. These voices have different emotional tones, and they hang around, mainly around your head, and is usually for some people just part of what they're thinking, is how they perceive them. But they will vanish. Cords will vanish. That's where you can, in this picture, see how you activate each other as you're standing there. If you have this infection, both of you, and give these feelings that you're connected with a cord. It also gives a sense that you sense the other's personality. Then there's tribal block. I do what my family or my culture expect. You can just imagine you're standing there and all your culture or your family is around you, and it limits what you want to do, because in your body that feels like all this emotion that they're radiating is coming through to you through the navel. This process also eliminates background noise in the head, so it's a really cool process. And this process, when we do it with psycho-immunology, just seems to be doing a much better job. It's deeper, or cleans it up better than our old process. Then we have manipulation fungus. The marker of that is that you can feel like there's a threatening presence around you, or you have a tiredness in a body area. So if a person feels threatened by the feeling of a presence, and this is what I have colored here in the dark, it can be behind you, it can be in front of you, or any place around your body. So, but if you feel like someone is there, you might have an activated manipulation fungus. And this manipulating feeling is coming from the manipulator via this fungus in your system. And it can be positive. It can be love, admiration, or it can be very negative, like hatred, anger, or evil, threatening. So eliminating this fungus also eliminates that feeling in the present, if you have one, that person disappears. And it also removes the vulnerability to be manipulated in this way. And other symptoms that this problem can cause is tiredness in a specific place on the body. That's if this one is really clumped onto the body in certain areas. It feels like a blanket. It gives a person anxiety or fear. So if you have a client, it can, or if you're just a person walking around, if you're very anxious, you might have one of these. It's not, there's so many things that can give anxiety and fear, but it's just one of the causes. We've also noticed that therapists or clients can have difficulty healing if they have one of these hanging on their, somewhere in their surroundings. So a really good one to get rid of. Now we have sucking holes. I have to be the center of attention. It's a good hallmark of that one. So this psycho-immunology process eliminates a pathogen, and it also repairs the center axis of the body. And this pathogen creates these sucking sensation. If you look at the woman here, it's along the center axis, and it can be in any, these holes, the feeling of them can have different shapes, different size, can be in different areas. They create this feeling of neediness, and it can be expressed like a craving or attention or love, addiction to love, want love, take things personally, just simply neediness, or insatiable, how do you say that? Insatiable, desperation, anxious if alone, feel deeply flawed, and it's their fault, my needs are not met. Or they can be energy vampires, or they can feel that their energy is just totally emptied out of them if they're around an energy vampire. So it's sort of the opposite pole of that. And once that's healed, these holes disappear, and the last part of this process actually also repairs the center axis, which gives a sense of completeness and solidity inside oneself. So another really great process. We see this in lots and lots of people, so it's a very common problem to have. Now we have the fourth one, copies of someone else's emotion. So if you feel that you're having an activated feeling, trauma feeling, but if you sense into it and it doesn't feel like it's actually yours, it could be that it's a copy. These copies feel like they are actually like little balloons stuck onto your body. And it's caused by a bacterial infection that actually, when we look at the trauma strings, are kind of stuck onto the ribosomes of the trauma string, giving the sensation of the overlap with the body there. Eliminating them can really be dramatic if a person has lots and lots of them. It's hard to say ahead of time, but so far in our testing, we've seen that people feel like a lot of negative emotion has vanished in their life, filling them actually with joy after following the process. Okay, soul loss. Soul loss can manifest as I miss someone or someplace. And a soul loss is this deep sadness, loss or longing for some place or something precious. And they feel like there's scooped up areas, like you're missing part of your body. And it makes people have weird behaviors, like if you want to track down whatever you're missing, could be an ex-partner or a place, some place that you're longing for. So healing is really a good thing. And you can do this with this process, with healing every single one of them at once. Then we have something called the pine cone fungus. This gives rise to 3 very different problems. So if you eliminate the pine cone sits in a structure inside the center of the cell, and it looks like a pine cone, but it's reality a fungus. It's totally packed, but there's some things that look like bubbles. That's why we call it bubbles. Some of them are ejected out of the pathogen and are circulating around in the area. some people actually move their awareness into one of these when they're stressed, and that can give this sense of suddenly feeling incapacitated. Other things that this fungus contributes to is time loops, which means if you have these and you heal a trauma and this trauma is inside a time loop, you can reactivate the trauma as if you haven't healed it. So the trauma just came back. And then there is the e-hole. If a person says, I just can't stand, I mean, this person, when I have interactions, I feel this evil place inside them. And that's actually because it's a match of what you have inside your body. So exactly the same location. So all of these will, disappear after healing the pine fungus. And that's also a really good one, because especially this time loop can bother some people a lot during their trauma healing. So what's next? Well, we in the research team, we are very excited about these 6 processes. When we're finished safety testing them, we will eventually make them available for clients, first in our clinic and later as certified therapists gets to do them also. And they're very effective. Instead of healing one by one, we can now make giant steps in improving people's life. And I'd also say this is, from a research perspective, the biggest one for me is safety testing. These processes have paved the way for psycho-immunology treatments for major medical conditions. So I'd like to end this and invite Grant and Mary to come on by saying acknowledgement to our applied research team. They have been working hard. This is Amy, Gitan, Julian, Lila, Leif, and Tomek. They have been testing our research protocols in our phase 1 clinical trials to test for safety and efficacy, and they've helped refine the processes to make them simpler. And as part of this, or spin-off, they've been, in addition, this team has been invaluable in our phase 2 clinical trials. They have simply been supporting the process of moving clients, our certified therapists who have participated in the phase 2 clinical trials safety testing. And I will just mention here quickly Agatha, Elisa, Eric, Astrid, Celine, Daniel, Danuta, Edward, Georg, Rita, Carolina, Kasha, Cryon, Matilda, Matthew, Natalia, Nikolai, Owen, Simon, and Sonia. Thank you, everyone. Without you, this would not have happened. And now it's time for questions.
Yeah, I just want to talk a little bit. You acknowledged our applied research team, which is amazing, and just is like, oh, thank you so much for being our phase 1 testers. But in our phase 2 clinical trials, as Kirsten said, our I think of them as mine, applied research team are working as therapists for doing this. For doing this phase 2 clinical trials, which is an incredibly huge amount of work. And all of us are doing this on a volunteer basis. But if you think about what needs to happen in those phase 2 clinical trials, we need 1 therapist for each test client. We are doing rule of 3, so we need to do it repeatedly to check, which means roughly 6 to 8 sessions for each person for each of these processes, and then multiply that by 12 to up to 20 people. The numbers start to add up really quick, the hours of doing it. And so it's been a huge labor of love for all of these people, for all of us. But it is a huge amount of work, but so much learning. And really what our applied therapists are doing is they're not only testing for the safety and efficacy, but learning how to do these processes better and better. And what are the words to say and those little tweaks? Those are the things that are like a side benefit and the little bonbon in the cookie box, the extra icing on the cake that we're learning from doing the phase 2 clinical trials. So it's been, first of all, this way of doing the safety testing has been a really good idea. the way that we're doing it. And so I just want to thank everybody, and we'll keep on going. That's all I wanted to say, Kirsten. Good job on the presentation.
Thank you for watching. For more information, please visit the website www.pigstates.com.